In this lecture, we're going to take a look at the connections and contrast the sympathetic and parasympathetic branches. And so with both the sympathetic and parasympathetic branches, we start with the origin of these connections from the central nervous system. And so the three examples up here on the top part of the slide illustrates the sympathetic nervous system, while the bottom will illustrate the extensions of the parasympathetic nervous system. And so if we begin with the bottom, we see that the parasympathetic nervous system will work first through preganglionic, what are described as cholinergic types of neurons. And a cholinergic neuron is one that will release a neurotransmitter that fits the classification of one that is cholinergic. And in this case, it's acetylcholine. And it will synapse onto postganglionic neurons, which are responsive to a cholinergic type of neurotransmitter. These receptors, so the neuron itself is referred to as a cholinergic neuron. However, the protein receptor that is sensitive to acetylcholine is considered a cholinergic receptor because it responds to acetylcholine, but it's also classified as nicotinic, that under experimental conditions is one that will bind the chemical nicotine in an experimental situation. The postganglionic cholinergic neuron will then release acetylcholine. Because it is a cholinergic neuron, it will use acetylcholine as its neurotransmitter, and the type of receptor on the effector cell for the parasympathetic branch is described as a muscarinic cholinergic receptor, that under physiological conditions, it's a receptor that will bind a cholinergic type of compound, in this case acetylcholine, but under experimental conditions will bind a chemical called muscarine. If we move to the sympathetic nervous system, we began by examining short preganglionic neurons that will extend into the close proximity sympathetic chain. And these preganglionic neurons are cholinergic in the sense that these are neurons that will release acetylcholine that will be detected by the postganglionic neurons, which are considered nicotinic cholinergic, which is to say that the cell bodies of these postganglionic neurons will contain protein receptors sensitive physiologically to acetylcholine, and under experimental conditions are described as being nicotinic because they can bind, these are receptors that can bind nicotine in terms of the receptor. The postganglionic neuron of the sympathetic nervous system is considered to be adrenergic. And by being adrenergic, it will release an adrenaline type of neurotransmitter. And the term adrenaline refers to a general class of compounds, one of which is norepinephrine. And norepinephrine will have an effect on effector cells that have protein receptors sensitive to norepinephrine. And these are just generally defined as adrenergic receptors. The long neurons of the sympathetic nervous system that directly connects to the adrenal medulla are defined as cholinergic. And being cholinergic, these long preganglionic neurons will release acetylcholine as the neurotransmitter that will trigger the adrenal gland cells, the chromaffin cells, to release epinephrine. And these are defined as adrenergic receptors. So the proteins embedded in the membrane of the chromaffin cells will be sensitive to an adrenaline type of, I'm sorry, on the effector cell will have proteins that are responsive to an adrenaline type of molecule, which is epinephrine. However, the protein receptors on the chromaffin cell are considered cholinergic, as these receptors respond physiologically to acetylcholine and experimentally to nicotine. The preganglionic neuron, the third type of extension from the sympathetic nervous system, involves relatively longer preganglionic neurons, which are also cholinergic, in that these are neurons that release acetylcholine that will be detected by receptors that are nicotinic cholinergic. And this is on the postganglionic neuron. So the postganglionic neuron houses receptors sensitive to physiological acetylcholine and experimental nicotine, and are adrenergic in that these postganglionic neurons will release norepinephrine and will be detected by protein receptors embedded in the membrane of effector cells that are sensitive to norepinephrine. Nicotinic cholinergic receptors are considered to be ionotropic, which is to say that these types of receptors that respond to acetylcholine will tend to cause cation channels to open. And so these are receptors that typically exist in channel form, in that when they are triggered by acetylcholine, will open up and allow the flow of sodium and calcium into cells. Muscarinic cholinergic receptors are considered metabotropic, indicating that these are receptors that, when triggered by acetylcholine, will activate internal metabolic processes. And these work through what are called G protein coupled receptors. G proteins are those contained inside the cell that are important for intracellular signaling. And so the activation of these types of cells will not cause the flow of cations, but will instead trigger an increase in certain metabolic pathways inside the cell. And the effect certainly depends on the type of cell. Autonomic postganglionic neurons are typically nicotinic cholinergic, where the effector organ for the parasympathetic branch is considered muscarinic cholinergic as well, while skeletal muscle is considered to have receptors that are nicotinic cholinergic. And so, as discussed in an earlier unit, skeletal muscle cells are sensitive to acetylcholine, or rather the protein receptors are sensitive to acetylcholine, making those receptors physiologically cholinergic type of receptors. In terms of the adrenergic receptors, these are receptors that are sensitive to the adrenaline types of transmitters. And all adrenergic receptors are coupled to the general class of proteins located on the interior of the cell associated with the cell membrane called G proteins. And there are different types of G proteins. There are what are called alpha receptors, defined as alpha 1 and alpha 2, and there exists what are called beta receptors, beta 1, beta 2, and beta 3. The properties of the alpha adrenergic receptors are such that they are located in the effector organs that are controlled by the sympathetic nervous system. And so when these receptors are activated, they are typically excitatory, and that being excitatory, that these receptors will tend to trigger an increase in a particular physiological or metabolic pathway in that cell. And these alpha receptors tend to have a greater affinity for norepinephrine than epinephrine. The beta 1 adrenergic receptors are typically found in cardiac muscle cells as well as kidney cells, and these are considered excitatory. So when adrenaline type of neurotransmitters bind to these receptors located in cardiac cells and kidney cells, these will tend to trigger an increase in metabolic activity of these cells, and they have an equal affinity for both norepinephrine and epinephrine. Beta 2 adrenergic receptors are typically found in some blood vessels and smooth muscle. And so by affecting smooth muscle, these types of receptors will often trigger a relaxation of smooth muscle, and as such are usually inhibitory. And so these types of receptors have a greater affinity for epinephrine than norepinephrine. These beta receptors are typically located in adipose tissue as well and are usually excitatory. Adipose tissue is made up of adipose cells or adipocytes, and these are the storage cells for triglycerides, which contain fatty acids. And so when norepinephrine or epinephrine, basically the adrenaline hormones, uh, bind to receptors, these adrenergic receptors that are in the membranes of these adipose cells, it will increase the breakup of stored triglycerides. In terms of the regulation of the parasympathetic and sympathetic nervous systems, the parasympathetic system is typically active under resting states and will tend to decrease heart rate and will tend to have an effect on reducing blood pressure. Whereas the sympathetic activity is opposite and is often described as that branch of the autonomic nervous system responsible for the so-called fight or flight response. And this heightened activity of the sympathetic nervous system is what is typically active and mediates preparation for any perceived emergency situation, physical or mental stress, as well as physical exercise. Some of the effects exerted by the sympathetic nervous system is an increase in heart rate and an increase in blood pressure. Because activation of the sympathetic nervous system will increase the secretion of adrenaline types of hormones, these hormones will have an excitatory effect on various cells in the body that will uh, mobilize energy stores. In other words, these are effects that will increase the breakup of fuels that such as stored fatty acids and stored carbohydrates in muscle and liver cells, and will also decrease gastrointestinal and urinary functions. It doesn't completely zero out GI or urinary functions. The focus here is on activating the cardiovascular system to support heightened metabolic activity, as well as supporting the activity of skeletal muscle cells. And this final slide will just review some of the main locations in the central nervous system that is responsible for many of these autonomic functions. The hypothalamus is considered the true master gland that will release various signaling molecules that will cause a wide range of downstream effects on human physiology. Just inferior to the brain is the brainstem region that is composed of the superior located enlargement called the pons and the medulla oblongata. And unique about these areas is that they house what are called respiratory centers. These are clusters of neuron cell bodies that will control the muscles that control breathing. And there are other areas here that house clusters of cell bodies that are called the cardiovascular centers. And so these are centers that are involved in regulating activity of the heart and of the blood vessels. So this ends this unit on the autonomic nervous system.